Welcome back to Intro to Physical Anthropology. I'm David Leitner. I'm your instructor. Today we're going to talk about Gregor Mendel. We're going to talk about the experiments he did on peas in the 1850s and 60s, and what he discovered about the patterns of the inheritance of traits from doing this. This is information that is foundational to genetics. Interestingly enough, wasn't really discovered by most geneticists until the late 19th century, and it is possibly, as we'll find out in another video, some of the strongest evidence for to support natural selection as a mechanism of evolution. So without further ado, let's get started here. Gregor Mendel was a monk in Austria. And, monks do a lot of other things than just sit around and pray. They oftentimes have hobbies or are interested in learning things. So, one of the things he did was to perform experiments on pea plants. He was interested in understanding heredity, that is, how are traits transmitted from one generation to the next. He originally did his research in the 1850s and 60s, interestingly, around the same time Darwin published On the Origin of Species, but he published in an obscure Italian journal. We didn't have computerized databases back then, so unless you spoke Italian and, had access to that journal somehow, if you lived in the English-speaking world or really anywhere else in Europe, you weren't going to know about his research, okay? So it wasn't really rediscovered until the late 19th century. Why is this even important? Well, his experiments concentrated on controlling for which traits he wanted to see. So he was looking at 7 what he called dichotomous traits. A dichotomous trait is a trait that only has 2 versions. Okay, he looked at seed coat. Was the seed round or was it wrinkled? Seed color, is it green or yellow? Shape, pod shape, full or constricted? Pod color, green or yellow? Flower color, violet or white? Stem form, axial or terminal? And finally, stem size, whether it was a tall plant or a dwarf plant. Okay. Overall, he ended up conducting experiments on 8,023 plants. 8,023. That's a lot. That's a pretty big sample size he's got there. So a lot of his results were really interesting and really reliable. Now, remember, he knows nothing about chromosomes. He knows nothing about genes. He knows nothing about any of this stuff we've been learning so far. So he is trying to figure this out without knowing anything about the mechanisms of heredity, the mechanisms, how genes transfer or how they create traits and that sort of thing. Now, there's a tool you can learn more about. I'm just going to go over it quickly in here. There's a video below on the Canvas site that does a better job of introducing you into Punnet Squares and Dihybrid Crosses. I don't highly suggest. You're going to watch it. And it teaches you how to do them. And this is important because essentially the Punnet Square mirrors the process of meiosis and the formation of sex cells. That's what we're doing when we draw a Punnet Square. And the idea is that along one end of the Punnet Square here, you've got one parent, and along the other side, you've got another parent. We list each of their genotypes in each box. So in this case, the mother and the father both have a mixed genotype. Capital B, lowercase b. Okay? And then we fill out the box. In the middle here, you've got one individual whose homozygous, that is, that has the same version of the allele for the dominant trait. So we call it homozygous dominant. We have 2 heterozygous individuals, that is, they have a mix of dominant and recessive. And finally, homozygous recessive, 1 homozygous recessive individual. Okay, this is a direct result of the process of meiosis. Mendel did experiments in such a way that he essentially was doing Punnet squares. Okay, he started off with what he called true breeding individuals. So there was this says F1, but there was an F0 generation where instead of having 2 mixed individual seeds, he had 2 individuals he knew were true breeding. That is, every time you bred them with another true breeding individual of the same type, they would produce the same type. So they were faithful from generation to generation. So one of them would have been homozygous dominant, and the other would have been homozygous recessive. And then when you mate them together, all of the offspring will be mixed. Heterozygous. As you can see here, you have a capital G and a lowercase g. Well, when you set up the Punnett square, you're just kind of mirroring this process here. You put the capital G and the lowercase g, and just like in the other one, we end up with 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Notice the recessive trait only shows up when you have 2 of the recessive allele. And so the genotypes end up being 1 homozygous dominant, 1 heterozygous, 1 homozygous recessive. So you get a ratio of 1 to 2 to 1. The phenotype, on the other hand, looks different. The phenotype of the dominant one is the only one to show through in the heterozygous individuals. So if we're just counting the phenotype, which in this case is seed color, we'll count 1, 2, 3 of the 4 individuals are yellow, and 1 of the 4 is green. And so we have a ratio of 3 to 1. Isn't that interesting? Again, just like we saw with the ABO blood system, the number of phenotypes and the number of genotypes don't necessarily match up. The most interesting thing is, here are the actual ratios he found when he did his experiments. All of them are very close to 3 to 1. All of them. Considering the conditions he was working with and not knowing anything about chromosomes and, not necessarily having great controls on the original true breeding population, he ended up with slightly different numbers for each of these, but they all hover around that 3 to 1 ratio because these are dichotomous traits. You're either going to be one or the other, and they are always going to, when you mix 2 heterozygous individuals together, you're going to end up with this pattern. Okay. Well, through doing these experiments, he came to 4 postulates, 4 conclusions he thought were probably going to be true no matter what. First was sort of definitional. He says hereditary characteristics are controlled by particulate unit factors that exist in pairs in individual organisms. I'm going to go deeper into the idea of particulate unit factors and particulate inheritance in another video, in the next video, called blending inheritance. But essentially what he is saying is heredity, instead of being like pouring 2 different colored paints into a paint can and mixing them up, it's more like pouring your red M&Ms and your green M&Ms into a paint can. So in other words, each of those M&Ms is a particle of inheritance that gets contributed. So he's starting with particulate inheritance. That is particulate 1. Second postulate. When an individual has two different unit factors responsible for a characteristic, only one is expressed and is said to be dominant to the other, which is said to be recessive. So here he's defining dominant genes and recessive genes, and he's saying dominant genes will be expressed whether you have one or two of that version of the unit factor. Recessive genes always require two of the same version. His third postulate. Now these get a little more difficult to understand, but I think with visuals it's a little easier. The third postulate is Mendel's law of segregation. During the formation, and this is not about segregation like in the South during the Jim Crow era in the 1960s up through the 1960s. The segregation in the sense that segregation just means to separate things. Okay. So during the formation of gametes, the paired unit factors, right? So they come in pairs, just like chromosomes, separate or segregate randomly so that each sex cell receives one or the other with equal likelihood. So if you we'll come to the next page here, if you look at the on the left-hand side here, at the top bubble on the left-hand side, the chromosome with the long red one and the long red with the blue tip, they are a pair of unit factors, right? And they are going to split up, and one's going to go into one gamete and the other's going to go in another gamete. And as you can see from the picture, the long red one went to the left and the red with a blue tip went to the right. Each of those had a 50-50 chance of going into either gamete. Okay, that's all this postulate says, that it's random chance which one goes in there. Okay. His fourth postulate, and this is what we're doing when we do a Punnet square, by the way. So each allele is sort of, we're representing the statistical distribution. Because it's a random process, it's statistical. We can come up with a statistical prediction of how many of the offspring will look certain ways. And there are only 4 combinations because there are 4 gametes that come out of meiosis. Mendel's fourth postulate is the law of independent assortment. So, during gamete formation, those segregating pairs, that is, those pairs of unit factors that are splitting up, assort independently of each other. Okay, let's go to the blow up again. What this is saying is, whereas the law of segregation it talks about a single pair of unit factors splitting randomly into either zygote. What this is saying is that the partners in 2 different pairs are no more or less likely to go into one gamete either. So each pair is going to split randomly. Okay? So just because long red here goes to the left does not mean long red in the medium-sized chromosome is going to the left, right? It's again, every single one of these has a 50-50 chance. All right? And that's important. Because that means some traits will travel together and some traits won't travel together. If they're on the same chromosome, they will travel together into that gamete. If the 2 traits are not on the same chromosome, then they could end up in different gametes. Okay, that is relevant to dihybrid crosses. Again, this looks very complicated. The video does a very good job of breaking this down. It's just being systematic about it. I will let the video instruct you on how to do that, but be aware as you're watching it, again, this is a direct reflection of meiosis. That is what we're looking at here. It's just that this is an extension of Punnet square. Instead of 4 possibilities, we have 16 because we're looking at 2 traits. Okay. Finally, linkage and crossing over. Linkage is just an expression of how close together two genes are on a chromosome. If you've got gene A and gene B, they're right next to each other. When crossing over happens, right? So that during meiosis, remember, part of the tail of the chromosome can flip and switch places with another one. The chances, there's only one place on that chromosome where you can split these two genes apart so they don't travel together. Okay? Because it's A and B. They're right next to each other. Whereas if you've got gene A and gene G over here, can split them up at like 5 different places in between. You can split them at B, C, D, E, and F, right? So all this is saying is that the closer together two genes are on a chromosome, the less likely it is that the more likely it is that they will cross over together. Okay? In other words, they are tightly linked. Whereas the further apart they are, they're more loosely linked. Crossing over, of course, is the thing that makes the independent assortment of linked genes have any effect whatsoever. Okay? Otherwise, if this didn't happen, you would just have the same genes, the same chromosome, each time. You would have two sets of two gametes that would be identical. There'd be no mixing up, really, whatsoever. So what crossing over does is it increases variation, because it, as you notice here, on the top here, we've got chromosome A, we've got the blue chromosomes with a dominant A and a dominant C. We've got the red chromosomes with a recessive A and a recessive C. If we don't do the crossing over in these next two steps, in the final step, we're going to end up with exactly what we started with. Dominant A, dominant C, recessive A, recessive C. But instead, we end up at the bottom, because of crossing over, we end up with dominant A, dominant C. Dominant A, recessive C. Recessive A, dominant C. And finally, recessive A, recessive C. Again, it's the Punnet square. This is sort of the same distribution here. You've got a mix of dominant and recessive traits here. So this is why crossing over is important, because it gives us more variation. All right, I'm going to end it there. Next time, we're going to talk about blending inheritance, and like I said before, about particulate inheritance, and how these two competing ideas were important to the theory of evolution, theory of natural selection. Until next time, take care of yourself, and have a great day.